Hello, my name is Dominic Roberts. I'm the Product Marketing Manager for GC Orbitrap at Thermo Scientific, based in the United Kingdom. I'd like to welcome you to the presentation today and to introduce our speaker. The presentation is titled A New Approach to Breath Biopsy Using Thermal Desorption Gas Chromatography coupled to Orbitrap Mass Spectrometry. The presentation today is going to be given by Adi Malkar, who is a Sales Development Manager at Alstone Medical in Cambridge, England. Adi's working currently on the breath biopsy platform with GC Orbitrap, and he holds a PhD in non-invasive metabolomic biomarker discovery from Loughborough University. You will have opportunities to ask us questions at the end, but for now I'll hand the presentation over to Adi, and thank you and enjoy.
Thank you very much for the introduction, Dominic, and thank you everyone for tuning in. Here is an outline of today's talk. I will start with a quick introduction to Alstone Medical and our breath biopsy technology. I will aim to highlight a few technical challenges in breath analysis. Then I would like to focus on discussing how we tackle these challenges in the analytical workflow using the Orbitrap. And I would like to finish by describing a case study which is on using breath biopsy for liver health. At Alstone Medical, our company's mission is to save 100,000 lives and $1.5 billion in healthcare costs. We're trying to do two main things: use our breath biopsy technology to develop diagnostic tests, and also provide the enabling technologies to the research community. So we mainly work with academics and pharmaceutical companies. We spun out of the Cambridge University in 2004 and have over 15 years of experience in volatile compound analysis in a range of industries. We're headquartered in Cambridge with presence in the US, and our multidisciplinary team is over 160 strong. Today, our breath biopsy technology is in use in over 100 clinics globally, and we have built the world's first high-volume breath biopsy lab in Cambridge. So the main question is, why analyze breath? Every breath contains thousands of volatile organic compounds, or VOCs. In the following set of slides, we'll look at what are these VOCs and where do they come from, and also look at why are they promising non-invasive biomarkers for everything from cancers to infectious and inflammatory disease. We focused on two fundamental challenges in healthcare today. First is early detection. So that's detecting diseases earlier when better treatment options are available and more lives can be saved. And second, enabling precision medicine. So this is to ensure the right patient receives the right medication at the right time. So which of these VOCs are present in breath and where do they come from? The thousands of VOCs on exhaled breath are fundamentally metabolites produced by the body. They come from two main sources: endogenous metabolites and exogenous ones. The endogenous metabolites are reflective of body's metabolism or perhaps from disease activity. These are the end products of gene transcription and protein expression. While our genes can tell us about the risk of getting a disease, it's the downstream metabolism that gives us a snapshot of what's actually happening in the body now. The other source is exogenous metabolites, which come from diet, smoking, medication, and also from microbiome from the gut or from the microbiome of the airways. All of these together mean that our VOC profile in our breath reflects our entire phenotype, providing detailed insight into how our body is functioning. So when the body's functioning changes, these VOCs on breath are well suited to detect such changes early. So the other question is, how do the VOCs get onto exhaled breath? Some are produced directly from the airway tissues or the lungs, but most VOCs originate from the bloodstream. The lungs are incredibly efficient at exchanging VOCs between the blood and the alveolar air, and in turn the exhaled breath. So this means we can collect and analyze VOCs pertaining not only to the airway diseases, but also systemic ones. The other important factor is that breath biopsy platform samples breath over a period of time. This allows for pre-concentrating the VOCs over time, which in turn drives sensitivity at the time of analysis. This figure summarizes a few key diseases for which potential VOC biomarkers in breath have already been reported in literature. These examples include both types of applications, the detection of disease as well as tracking disease progression. This list includes various cancers, cardiovascular diseases, inflammatory diseases, as well as metabolic disorders. This highlights the relevance of breath biopsy for a wide range of diseases. In order to identify really robust breath-based biomarkers, which can be introduced into the clinic, breath analysis needs to be incorporated into clinical trials. One of the challenges in the past has been how do you collect a high-quality breath sample in the clinic itself? This is where our receiver breath sampler comes in. It was developed to enable patient breath samples to be collected reliably and reproducibly, making it ideal for large clinical trials where patient recruitment and sampling can span across many months. During sampling, patients just breathe normally while the receiver tracks their breathing pattern and captures a specific volume of breath and selected breath fraction. Over a period of time, receiver traps and stores the VOCs and pre-concentrates them onto thermal desorption tubes. Once collected onto these tubes, the VOCs are stable and can be shipped at ambient temperatures to our breath biopsy lab for analysis. For each sample collection, we provide a conditioned and QC-checked mask and thermal desorption tubes or the cartridge. The receiver has been designed with patient safety and comfort in mind. It is also CE marked. So along with the receiver, we use the Casper portable air supply, which is designed to allow the receiver collect a higher quality breath sample. It provides a clean VOC-free air to the receiver, which the patients can breathe in, and this helps to minimize the background contamination. There is also our breath biopsy collect software, which controls both the receiver and the Casper systems during sample collection. So what else do we need to identify and validate biomarkers from breath? The answer is an entire platform that is reproducible, sensitive, and robust. We also need end-to-end workflows right from sample collection through to analysis. The whole process starts with installation of the receiver at the clinical trial site and regular supply of consumables to enable sample collection. Once the samples have been collected by the clinicians, the thermal desorption tubes or the cartridges, along with any subject metadata relevant to the study, is transported back to our breath biopsy lab for analysis. Again, these cartridges are shipped at ambient temperatures, so quite a straightforward shipping process. Once we receive the samples, we run a series of checks on these for any errors during sample collection before they are analyzed using our standardized analytical methods using TDGCMS. And then this is followed by comprehensive analysis by feature extraction and statistical analysis combining with the study metadata. This data is then interpreted by our clinical sciences team before compiling a report at the end of the study. What I wanted to focus on today was the incorporation of thermo-Orbitrap into our breath biopsy analytical workflows. The way in which we collect breath samples using the receiver, that is by pre-concentrating onto thermal desorption tubes, means that we collect a high abundance VOCs along with trace level ones from breath. Breath also contains a wide range of analytes as discussed before. As a result, we need analytical equipment that can cope with such complex sample analysis. So we rely on TDGCMS as the gold standard for breath analysis. We've started a collaboration with Thermo Fisher Scientific, and we have upgraded our instrumentation by integrating the Q-Exactive Orbitraps into the Alstone Medical's breath biopsy platform. This upgrade has the potential to advance the applications of our non-invasive breath sampling by addressing some of the well-known challenges in breath analysis. The inclusion of the Orbitrap is enabling quite a few interesting changes to our workflows. One of the new additions is a dual analysis approach, which is to do a targeted analysis alongside our discovery or untargeted analysis. This is unlike our earlier workflow when we had to choose between one of the two approaches. So the initial parts of our workflow remain the same here. That is, samples arrive at the breath biopsy lab and we create a run sequence using our in-house samples database. But then we can run untargeted as well as a targeted analysis on the same sample at the same time. The benefits of that is that we can have a targeted panel with absolute quantitation as well as perform a discovery analysis to look for those novel biomarkers and produce feature lists that can go on to statistical analysis and provide data for clinical interpretation. And then the results from both of these approaches can be combined into a single study report. The inclusion for targeted analysis also enables the performance of the instrumentation and that of the breath sample collection to be quantified. So let's start looking at the challenges in breath analysis. The first one, which is a very obvious one, is the complexity of breath as a sample matrix. Here, I'm not going to talk about the sample collection or the clinical challenges, but the analytical challenges of actually analyzing breath samples on TDGCMS. As we've seen so far, the breath samples that we collect are complex. There's a lot of information captured on these thermal desorption tubes, and typical concentrations of different chemicals or analytes can easily span 4 to 5 orders of magnitude. So we need to be able to detect and measure high abundance VOCs as well as the trace level ones in a single analysis. This means that our analytical methodology needs to keep up. When we are developing the analytical methods, we need to typically try to balance the requirements of selectivity, sensitivity, and the scan speed of our mass analyzer. Here, the introduction of the Orbitrap allows us greater control over mass resolution. The AGC target prevents overloading of the mass analyzer and in turn allows us to measure high abundance ions and not to overwhelm the detector. It also allows us to maximize the data scan rate, which is great for the deconvolution and feature extraction, which is of quite a bit of an importance in untargeted metabolomics. High resolution accurate mass is crucial for metabolomic analysis and identification of chemicals, particularly low concentration chemicals in complex matrices. Orbitrap gives us the ability to achieve sub 1 ppm mass accuracy across the width of a chromatographic peak. We can see an example of one of the VOCs here. It's 2,5-dimethylfuran. So we can see the mass accuracy for one of its ions across the chromatographic peak. This and the stability of ion ratio is crucial in spectral deconvolution, which is of critical importance in untargeted analysis. The Orbitrap also guarantees stable ion ratios even at high sample concentration, so this improves deconvolution, compound identification, and the subsequent generation of a custom library. The next challenge in breath analysis is untargeted discovery. Comprehensive analysis is an important aspect of the breath biopsy workflow for metabolomics and biomarker discovery. The mass accuracy from the Orbitrap systems guarantees that the samples that we analyze will yield good quality data fit for untargeted analysis. From this data, we need to extract features for subsequent analysis. The crucial part here is reliable and reproducible deconvolution of the raw data. Through our partnership with Thermo, we beta tested the new version 3.1 and 3.2 of the Compound Discoverer. The built-in GC deconvolution algorithm makes the untargeted part of our workflow possible. The new addition there was the incorporation of the deconvolution software right into the statistical analysis. So we have a single software for the entire untargeted approach. It also includes the option to embed custom scripts such as Python and R, and as a lot of our statistical analysis packages are built on Python and R at Owlstone, this feature was especially useful in streamlining our standardized analysis pipeline. Another important element for us was the ability to export a flat feature table, because it allows us to identify and track a feature from a sample across multiple samples and across multiple studies using unique identifiers. In the latest variant, the version 3.2 of the software, which we are currently beta testing, the deconvolution algorithms are tailored to different ionization methods. So, for example, for electron impact and chemical ionization. And both these workflow options can search ChemSpider database as well as carry out pathway mapping, which is quite useful for when we think about biological interpretation of the data. We have also seen improvement in processing speeds using the new algorithm when processing large batches of samples. The first thing which we wanted to do to assess the performance of the software was to verify the output of the deconvolution. So for this, we prepared chemical standards of a few target chemicals and we analyzed them, and then run the resulting data through the untargeted deconvolution software. So far, we've had a very high success rate. The software could deconvolve 32 out of the 33 chemicals that we analyzed. The one that we couldn't find was actually a perfect co-elution event, and any deconvolution software would have struggled to pick that up. So we're quite happy there. Deconvolution brings us to the next challenge in breath analysis, identification. The identification of unknowns from the untargeted analysis is what becomes important for clinical interpretation. Just seeing the abundance of a VOC go up and down between study groups isn't enough. You need to be able to try and interpret where that VOC came from and what biological pathway is causing that difference. We can do tentative identification via typical spectral matching, that is against a reference NIST spectra or from SI score. However, with the Orbitrap, we have another option. We can use the accurate mass to build high-resolution compound libraries, which can be searched against to find a hit. Compound Discoverer can then calculate high-resolution filtering, that is HRF scores, from high-accuracy EI data, where a match gives you high confidence that that feature is the ID that you're saying that it is. One of the benefits of this is that it allows comparison of compounds of interest that are identified across samples between studies. The accurate mass library gives you the option to say that was the same chemical on the same or a different day in different studies. This can also be combined with the NIST library. So we can use standard libraries alongside the high resolution libraries at the same time, meaning we can continue to offer tentative identification for VOCs that aren't in our accurate mass libraries yet. So a little more detail on how we create an accurate mass library on our breath biopsy platform. We first analyze the chemical standards on the TDGC Orbitrap. We use the deconvolution algorithm and compound discoverer to predict the fragment ions and theoretical masses to identify a compound. We add the acquired accurate mass spectrum to our custom breath biopsy library. And then we can use that back in software to say this compound from breath is compound X. In this case, it's 2,5-dimethylfuran. And here is an example of this working in breath. On the left, we have one nonene. Now, alkenes can be very hard to ID because they are very similar in fragmentation patterns. So it's often the ion ratio that becomes important in identifying them. And on the right is limonene. I'll come back to the clinical importance of limonene and why it's important to us in a little more detail later on. But it suffices to say that being able to identify this to a very confident level is very valuable to us. So that's some examples of how we're able to build and use high resolution accurate mass library in an untargeted way. But it can also be used for targeted analysis for quantification of breath VOCs. We can calibrate our target VOC and then extract the target from a complex breath chromatogram using our accurate mass library spectrum and a known retention time window as a reference, and then quantify the amount of that VOC on the breath sample. Stable implementation allows us to quantify it across studies, across days, and across samples. So based on all of that, we have started to build our very own breath biopsy compound library, which is expanding as we continue to analyze more standards. Here is a selection of compounds, and we can see that the range of compounds is evident in terms of volatility and chemical classes. Expanding this library to include additional biochemically relevant compounds is our current focus. This has the potential to vastly increase the confidence we have in the identification of VOCs found in our samples. This is a significant step and will soon allow us to carry out measurement of panel of VOCs which can be associated with a particular application or disease area. So going back to the topic of stable instrumentation, we have effectively a service lab. And so we need instruments we can rely on, which don't need continuous maintenance, which are capable of working in the high throughput environment. One of the biggest appeals of the Orbitrap from an operational perspective is that the ion source can be removed, changed, cleaned, all without breaking vacuum, which means a reduction in instrument downtime. The option of the dual filament is also great because if one goes down, we can still run the instrument until we need to change it. So it allows for completion of the analytical run. And one of the biggest game changers is our implementation of the Chromeleon Chromatography Data System, or the CDS. The software is important to us because it first and foremost is a data management system, and it is cloud-based. We also use it to control our instruments, manage our analytical processes, implement control of users. This ultimately provides integrity to our data and report and results all in one software. The instrument control through the CDS, which gives us the ability to control the Mark's thermal dissolver, the Thermo GC, and the Thermo Orbitrap in the same software, in a single sequence can't be understated. It's mostly for the simplicity it provides to our analysts. It also provides the system suitability checks at point of acquisition. So combining it with our targeted data processing, we can look for those targets, apply limits at point of analysis, and have those limits applied to real-time data. Alongside the targeted analysis with Chromeleon, we can also automate calibration and qualitative as well as quantitative data processing. We also have the option to run automated checks of our QCs throughout. So if something goes wrong in the sequence, it is automatically paused. This, along with increasing our sample yield, most importantly, preserves the precious clinical breath samples. This is really important when we remember that these samples are often collected from quite sick people and involves a lot of deliberation and effort in setting up. All of this combined is what then feeds back into the automated monitoring and the system suitability tests to protect our samples. Monitoring of those QCs so far has produced very good intra and inter-sequence variation. Here are two sequences which have been overlaid for the performance of Octane to show that even at absolute level, the response has been very stable. The quantitative accuracy as well has been so far greater than 95% in all our studies. Additional to the data processing capability, we have the things that help with running of our high throughput services lab. Alongside all of that comes a full audit trail, a complete instrument log, and user accountability. The system allows for user roles and permissions specific to the user roles. This ensures that user accountability when handling client breath samples. This ultimately ensures data quality control. For example, an analyst can process and submit data for independent review from a data reviewer. That reviewer can then make sure all the system suitability checks have passed and ensure data quality. This can then be sent to approval for the lab manager to guarantee high quality service and faster turnaround. Here is an example of a sequence audit trail. For every single sequence, a comprehensive audit trail is available. This is quite useful for ensuring high quality analysis and traceability of clinical breath samples. We're also able to create automated and customizable reports, which can include the audit trail, the qualitative analysis, and full output of the system suitability checks. This is very useful and helps us streamline reporting and management of client study data. So in summary, we have been making great progress at integrating the Orbitrap systems into the breath biopsy platform. The compound discoverer software is great at spectral deconvolution and hence feature detection, and this has improved our breath biopsy discovery analysis. By building our own breath biopsy compound library, we hope to achieve confident identification of unknowns, which will be invaluable for further clinical interpretation from our studies. The CDS software is streamlining our processes by unifying instrument control, data management, QC processes, and automated reporting. So as a culmination of all this work, the first breath biopsy services study using the Orbitrap began in January 2020, with wider rollout planned across 2020. Like most of us, we're feeling the effects of pandemic, but we are making the most of this time to optimize our breath biopsy platform, incorporating the Orbitrap. So for the last segment of this talk, I'd like to describe the assay for limonene using breath biopsy. As I mentioned earlier, one of the things we are keen on as a company is developing tests. So what follows is an overview of a concept and some early stage experiments for analyzing limonene from breath. One of the things I hope to achieve here is to give you a flavor of wider applications outside of untargeted biomarker discovery using breath biopsy. So why did we start thinking about limonene? All of the existing liver function tests, which are routinely used in clinics, largely rely on measures of liver damage and not of liver function. Even the imaging and liver biopsies used to diagnose chronic liver diseases don't directly reflect liver function. The absence of any non-invasive liver function tests slow down the drug discovery efforts in this space. It's been reported in literature that levels of limonene are elevated in breath of liver cirrhosis patients, and this has a potential to be a non-invasive breath-based biomarker for liver function. The pathway to chronic liver diseases often takes years to establish, and it involves different etiologies. In most cases, symptoms appear only at late stage. and often diagnostic methods are invasive. Our goal is to develop a non-invasive breath test which is able to detect NAFLD and NASH, which is non-alcoholic fatty liver disease and non-alcoholic steatohepatitis. A handful of studies compared breath of cirrhotic patients with that of healthy individuals and found limonene as a potential biomarker associated with cirrhosis. There's quite strong evidence of association of limonene in breath with liver. A couple of publications summarized here, particularly interesting. First, limonene was found elevated in patients with cirrhosis and levels normalized after liver transplant, linking levels of breath limonene to liver function. Second, stratification of cirrhotic patients by hepatocellular carcinoma, or HCC, showed that patients with HCC have moderately elevated levels of limonene compared to patients with cirrhosis. So why does cirrhosis increase in limonene? So why does cirrhosis increase limonene? So why does cirrhosis increase limonene on breath? Limonene comes from diet. So for example, from citrus fruits or flavorings. It is absorbed and metabolized by two liver enzymes, CYP2C9 and CYP2C19. Cirrhosis downregulates these enzymes, meaning that limonene accumulates, which leads to an increase in limonene on breath. We did a pilot study of a total of 84 subjects with these goals. So we wanted to look for proof of principle that a breath biomarker can be used to detect cirrhosis or HCC, independent validation of literature results, performance and correlation with blood biomarkers, and identify possible confounding factors. The samples were analyzed using breath biopsy platform, which is presented, including the Orbitrap. The inclusion criteria is shown in the table here. The controls do not have cirrhosis or HCC. The cirrhosis severity was assessed through Child-Pugh scores. The subjects were not asked to do anything specific prior to the breath collection, so the limonene intake varied. So here are our findings. There was significant increase in breath limonene for cirrhosis and cirrhosis with HCC groups above the control population. There was no significant effect of HCC within cirrhosis patients, so both groups were matched for cirrhosis severity. When cirrhosis is grouped by severity, breath limonene levels do show an increase with the disease severity. Limonene can detect cirrhosis, but seemingly not HCC. As such, groups were combined subsequently and can be seen on the right. We found no impact on limonene levels from a range of other confounding factors, including diabetes, portal hypertension, body mass index, etc. We did find a relationship between dietary intake of limonene and the resulting detected levels. Use of drugs that interact with the same liver enzymes also show an effect. Both of these are to be expected based on how limonene interacts with the body, as shown earlier. Using limonene to train a classifier resulted in this ROC plot, used to discriminate cases and controls. The optimal model is indicated by the Uden index, which provides the best possible balance of sensitivity and specificity, so true positive and true negative rates. This optimal model sets the limonene threshold for cirrhosis at 10.2 nanograms per liter. A larger study with controlled limonene intake may be able to produce a more accurate model with a different threshold. Additional studies could also relate limonene to earlier liver diseases that are more challenging to detect. A number of other biomarkers of liver health also exist. These typically aren't compatible with non-invasive sampling. So bilirubin from blood cells is processed by the liver for excretion and builds up in late-stage disease. Albumin is synthesized in the liver and falls due to liver damage. The liver also produces prothrombin needed for blood clotting. Hence, poor blood clotting, low INR, can also indicate liver disease. As cells in the liver die, they also release old enzymes into the blood, leading to an increase. As such, ALT is a biomarker more of liver damage than that of liver function. Limonene levels correlate with most other biomarkers, so bilirubin, albumin, and INR. Notably, these are all components of the Child-Pugh score, which I showed earlier. It also shows an overall relationship with limonene. Limonene does not correlate with ALT, the marker of liver damage. As expected, this indicates that limonene can be used to assess liver function. So in conclusion, breath offers an exciting approach for non-invasive disease detection and monitoring. There have been challenges, namely in reliable sample collection and analysis, but applications are widespread and quality results are emerging. The breath biopsy platform offers a versatile and robust solution for the collection and analysis of breath for a variety of research applications. The integration of Orbitrap mass spectrometry and the latest analysis softwares, combined with an accurate mass library, offers the capability for a reliable biomarker identification and monitoring. Targeted analysis of specific VOC panels is also possible for a growing selection of compounds. We have applied this technology to validate results indicating a relationship between limonene and liver disease and to demonstrate that this relationship correlates with other known liver biomarkers. So I would like to conclude here, but not before acknowledging everyone who's actually done all this work. I'd like to thank our collaborators from Thermo, in particular Christian, Julia, Dominic, and Jason. Also, a massive thanks to the Alstom medical team behind all of this work. Me and my colleagues would be happy to take any questions now, or please feel free to reach out to us through our website. And thank you all for listening.